Hello students, I am Amjad Fatmi, and in the last class we have discussed about the centrifugation. What is centrifugation? What are the common terminology or factors used or applied during the technique of centrifugation? Today we will discuss what types of centrifugation. What are the types of centrifugation? So there are basically two types of centrifugation. That is the preparative centrifugation, and another one is analytical centrifugation. Okay, preparative centrifugation is mainly concerned with the separation of cells or subcellular structures. And in the Mendelian cycle, what are the components present in the cells, within the cells, that are get separated through preparative centrifugation. Okay, this is the first step, because for it to study any component, it has to be separated from the rest of the components. Okay, so first, this is the first step. And in the analytical centrifugation, we study the physical properties of the biomolecules or macromolecules. Physical properties like their molecular weight, their shape, or their density, etc. So these physical property can be studied using analytical centrifugation, and by using preparative centrifugation we can separate these components. Okay, so we will discuss preparative centrifugation today. There are two types of preparative centrifugation. One is differential centrifugation and another one is density gradient centrifugation. Okay, in the differential centrifugation, the centrifugation separation is done by in a stepwise manner. Okay? In the stepwise manner, the centrifugal field is increased in a stepwise manner. The first minimum centrifugal field is applied. In the next step, a higher centrifugal field is applied. In the next further step, a even higher centrifugal field is applied. That means the centrifugal field is increased in a stepwise manner. That results in separation of a smaller molecule. First, the heaviest molecule is separated. In the next step, when the centrifugal field is increased, a smaller molecule gets separated. In the subsequent step, when the centrifugal field is even higher, the even smaller molecule gets separated. So that means that this is a stepwise manner or a stepwise process of increasing the centrifugal field, resulting in separation of smaller and smaller particles. Okay, so this is the process. How it is done? First, homogenates is taken. Homogenates means that all the components of the cell are present in a liquid medium, in a solution, and distributed evenly in all part of the solution. Okay, that is the homogenate. The first step is for the study of centrifugation is first forming the homogenates of that tissue which is to be analyzed. Okay, or contain that molecules or components which is to be analyzed. Okay, the first homogenates is of form of that tissue. So suppose this is the homogenate. This homogenate is subjected to centrifugation, resulting in sedimentation of largest particle. This is the largest particle sedimented, and the rest of the homogenate remains in the solution, and that solution is known as supernatant. The supernatant is the component of the homogenate which is not settled down or sedimented or present over the sediment. Okay? So this is the sediment, and in the next step, this supernatant is collected into another test tube. Okay, so that means that if this supernatant is collected in another test tube, automatically what happens? This component or sediment will be separated, get separated from this supernatant, or we isolate this sediment which contain some component of desired component, okay, to be studied. Okay, so this is the first step. In the next step, this supernatant is again subjected to centrifugation with a higher centrifugal field, okay, resulting in separation of component another component. This component which is settled down, is sedimented here, is smaller or than this component, or having a less density than this component, because those component which has largest size or maximum density will have maximum sedimentation rate, or will sediment first. This will separate first, the heaviest will separate first, and then in the subsequent step, this component which has less density than this one will get separated. And the next further step, suppose third step, this supernatant is collected in another test tube, that is test tube third, and subjected to centrifugal field of higher magnitude. Okay, then this one, this centrifugation field will be higher than minimum, this will be higher than this, and this will be higher than this. Okay, in this way centrifugation is carried out by increasing the centrifugal field in a stepwise manner, and resulting in the isolation of separation of smaller and smaller molecules. Okay, which we will discuss, described by this diagram also. Suppose this is the tissue, suppose this is a contractile tissue or muscular tissue. Okay, first this will be homogenized. Homogenization is done, or we'll get a tissue homogenate. This tissue homogenate is taken into test tube, and it is subjected to centrifugal field of 100 G for 10 minutes. We have already discussed about the G, capital G and G, and what is the relative centrifugal field. This is a relative term in terms of G. How many times of G is small g is applied. Okay? So the centrifugal field applied is about 100 G. Okay, for how many time? How much time? This will be for 10 minutes. Okay? This centrifugation results in formation of pellet 1. This pellet 1 and this pellet 1 contains nucleus. So that means that after centrifugation at 100 G for 10 minutes, nucleus gets separated from the rest of the homogenate. We collect the supernatant, this supernatant, and this supernatant is again subjected to centrifugation at 10,000 G for 10 minutes. This results in formation of pellet 2. This pellet 2 contains contractile apparatus if it is the tissue is a muscular tissue. Okay? Then this contractile apparatus is separated from the supernatant by collecting this supernatant into another form here, okay? And this supernatant is again centrifuged at 20,000 G for 20 minutes, resulting in formation of third pellet, that is the mitochondria. Okay? Okay? And this is the supernatant which is collected in another test tube, okay, and subjected to centrifugation, very high centrifugation of about 100,000 G for 60 minutes. That results in formation or separation of microsomes, okay, smaller membranous structure, okay, and these microsomes and the rest of the supernatant will contain cytosol. That means by